The previous research manifests the acute BHPF exposed even in 1 micromole per liter. How about the concentrations approximately aquatic environment, and if there are any drugs that can recover the effect induced by BHPF? In order to learn more about these questions, the exposed concentrations and the endogenous biological hormones melatonin was applied in further study. In this section, I will introduce the further study about BHPF on development and sleep-wake behavior. Previous studies have shown that acute BHPF exposure leads to early developmental malformation in a low concentration. The effect of a low concentration on zebrafish early embryonic development and larvae behavior is not yet known. Melatonin is an endogenous biological hormone. Melatonin can improve sleep quality, and it has important regulator effects on development, oxidative stress, DNA damage, and so on. In this section, we try to study the effect of BHPF dose close to environment water concentration on the early development period and sleep-wake behavior of zebrafish. At the same time, melatonin was used to investigate whether it can improve the development and behavior abnormality caused by BHPF exposure. The flowchart displayed the group setting, observation, and detection check windows. The treatment solution was refreshed every day. The morphological observation in pseudohybridization, immunofluorescence, surface tension, sleep-wake behavior, and qRT-PCR was conducted at different development periods. The results showed that melatonin eliminated development delay and mortality increase in zebrafish embryo induced by high-dose BHPF. In the figure, we can see that melatonin eliminated development delay improved the 100% epiboly rate at 10 hours post-fertilization and consequently increased survival rate under high-dose BHPF exposure. The surface tension between the control embryos and the treated embryos was an indicator of biomechanics. The alternation of surface tension indicated the change of embryonic coring features under BHPF exposure. As the figure described, there are 3 surface tension among the 2 embryos and the water media, and 3 angles existed between the surface tension. The results showed that the angles between the tenant left or right embryo water surface and the angle between the 2 embryo water surface changed under high-dose BHPF exposure. Moreover, the ratio of the right surface tension and the left surface tension represents the surface tension of BHPF treated embryos and the surface tension of control embryo. The ratio was obviously increased under high-dose BHPF exposure, while melatonin recovered the ratio of surface tension under high-dose BHPF exposure. In order to learn more about BHPF and melatonin on early development, the whole-mount-induced hybridization was used to show the spatial and temporal distribution of several key genes during the early development. Compared to the control group, the high-dose BHPF exposure weakened the expression of early development marker genes,
including NTL, SHH, KROX20, PAX2, and CMLC2.
Meanwhile, melatonin elevated the aberrant early gene expression. The embryos with melatonin treatment improved the aberrant expression patterns induced by high-dose BDPF exposure. Besides that, melatonin improved the typical morphology, including body length, head, and eye area, which was triggered by high-dose BDPF. To further explore the effect of BDPF on zebrafish locomotor activity, we observed sleep-wake behavior. The sleep-wake behavior test is an efficient way to study the effect of new drugs or compounds on nervous system. Figure A shows a simple flowchart of the behavior test. BDPF and melatonin exposure started from 2 hours post-fertilization, and at 96 hours post-fertilization, the larvae was transferred to a 96 well plate, a continuous 48 hours detection, including 2 dark and 2 light cycles, begin from 108 hours post-fertilization to 156 hours post-fertilization. As we can see, BDPF exposure increased waking activity and decreased sleeping time. Melatonin resulted BDPF-induced wake increasing and sleep decreasing in the larvae zebrafish. The motor neuron derives from the central nervous system and projects axons to the somatic muscles. Motor neurons can innervate the muscle fibers and then interface locomotion. To further learn the connection of alternation of sleep-wake behavior and neural regulation, the distribution of caudal primary motor neurons was studied by immunofluorescent staining. As the figure showed, BDPF exposure obviously truncated the neural axons, while melatonin reduced the damage of caudal prime neurons. Moreover, qRT-PCR results showed that BDPF exposure upregulated expression levels of sleep-wake related genes, including HCRT and HCRTR, in a dose-dependent manner. It indicated the activation of hyperokine-auxin pathway, while melatonin recovered the overexpression of HCRT and HCRTR signal caused by BDPF exposure. At the same time, we noted that BDPF exposure improved the expression of ENET2. ENET2 is the gene to code the key enzyme in endogenous melatonin synthesis. Although BDPF exposure upregulated the expression of ENET2, BDPF exposure alone did not increase melatonin levels. The melatonin levels increased only in the groups with exogenous melatonin addition. To investigate the regulation between the HCRT-HCRTR signal and ENET2, the HCRT receptor antagonist SUV and TCS were used to inhibit the hyperokine-auxin pathway. The result showed that inhibition of this pathway decreased the expression of ENET2 in control group. Similarly, ENET2 expression in BDPF group with SUV and TCS decreased significantly compared to the BDPF exposure alone. This indicated that ENET2 is the downstream of HCRT and HCRTR. This section shows that BDPF interferes with sleep-wake behavior by activating hyperokine-auxin pathway, induced insomnia-like phenotype, with the increase of wake activity and the decrease of rest, while melatonin acts on the pathway and improves sleep-wake behavior. Thanks for attention.